Okay, well, today I'll talk a little bit about the population genomics of the western chicken turtle. But before I do, I want to thank a whole bunch of people, right? Particularly all these brilliant students and postdocs that have worked in my lab over the last few years, as well as the collaborators on this particular project, which include Paul and Toby Hibbets and Wade Ryberg at Texas A&M, but especially Natalie Allen. Natalie's one of my PhD students, and this work really represents one of her primary dissertation chapters. And so she gets all the credit, and if there's any blame, that's mine. So hopefully I won't butcher her work too bad. I also want to acknowledge all of the agencies that have funded our work over the last few years, in particular because I'm going to give a couple of other non-chicken turtle examples. And so I want to make sure I've properly thanked all the folks who funded that work. But I especially want to thank all of you for attending today, and Texas Parks and Wildlife for funding this research and for working with us on this the last couple of years. So Paul may be a real herpetologist. I am not. I'm not a herpetologist in that we study lots of other organisms, all sorts of vertebrates, um, everything from whales to pupfish, and you'll hear about a couple of those other examples today. But what I do like to do is try and take advantage of all the information that's contained in genes and genomes to make inferences about organismal biology and especially how that relates to conservation and management. So, with that regard to whether we're working with prairie chickens, chicken turtles, whales, pupfish, uh, in general, we're working with biological samples that come from wild populations. Maybe it's a blood sample, maybe it's a tissue sample from a harvested animal, maybe it's a scat sample. But regardless, we take it into the lab and we don't get this stylized double helix DNA shown in this bottom blue slide or bottom blue photo. The one on the right is what the DNA actually looks like in the lab. When we get that, then we can do all sorts of analyses to try and understand the genomes. And so, for those of you old enough to remember a set of encyclopedias, I like to explain the nuclear DNA over here as sort of a set of encyclopedias, right? Where chromosome 1 might represent volume A in the set of encyclopedias, chromosome 2, volume B, and so forth. So there's a lot of information here, half of which comes from mom, half of which comes from dad. So you get one copy of chromosome 1 from mom, one from dad, one copy of chromosome 2 from mom, one from dad, and so forth. The mitochondrial DNA we only get from our moms, right? It's always maternally inherited. And it's a very different genome in many ways. And I think of this as more like a dictionary. So both genomes, the nuclear genome and the mitochondrial genome, can be thought of as this reference material for, um, organismal biology, but they're just different reference material. And so I'm going to skip all the details about how we get genome sequences, but on the far left here, you see a double helix that represents DNA. And within that, these colored bands in the very middle of that DNA represent these molecules that I'll just refer to as GAs, Ts, and Cs. These are the base pairs that make up the millions and millions, billions of nucleotides of GAs, Ts, and Cs. And then we use some lab procedures to try and determine the sequence of all those. As you could see here, the sequence in this direction would be T, A, C, T, C, and it goes on and on and on throughout the whole set of encyclopedias in the dictionary. And then we use a computer to try and put those all back together computationally and organize them in a way that hopefully looks like the actual chromosomes do in the organism. Okay, and why do we do this? Well, in part to measure genetic diversity through um, different lenses like heterozygosity. And heterozygosity is this idea that sometimes we get the same allele or the same variant from mom and from dad. So in this case, this individual at this chromosome got a big B from mom and a big B from dad. This other individual got the same two from mom and dad, but they're both little b alleles. So different, but there's no diversity within an individual, right? Whereas heterozygous individuals got either a big B from mom, a little b from dad, or a little b from mom and a big B from dad. And together, these two categories are called heterozygotes. And there is some genetic diversity at this locus. And you can imagine this spreading across all the loci on this set of chromosomes, but also across all of the chromosomes in the genome. So heterozygosity is one measure of genetic diversity, and it's one that a lot of people can relate to because uh, it relates to concepts that are familiar, like family trees or pedigrees. And so here we've got mountain lion populations shown because this is a famous example. And you can see that when we have outbreeding, right, if we have a heterozygote mated with the homozygote, then we get a heterozygous offspring. So there's some genetic diversity in this offspring. Whereas if we have inbreeding, so here this pedigree indicates these two individuals are full siblings, like a brother-sister mating. When this happens, we more often get inbreeding, so that we've got homozygous individuals that then can exhibit deleterious traits like malformed spines, right? Which is not a good thing if you're a mountain lion. So these are concepts that are pretty familiar to many of us. And so that's why I talk about heterozygosity as a component of genomic diversity. Well, today I'm going to talk about our recent ongoing work that's largely driven by management agencies like TPWD, but also federal or regional agencies and other studies. And I'm going to talk a fair bit about delineation, right, which is the process of identifying species or populations or what may be called evolutionarily significant units. Uh, Endangered Species Act, I think, uses the term distinct population segments. So it's just delineating what we mean by those different terms using genomic data. And I want to talk about a couple of examples, not with turtles, but with pupfish and prairie grouse, before we get to the turtle data to, one, explain some of the analytical approaches that we're using and make sure that everybody's got some appreciation of those, but two, also put the genomic diversity into context. Instead of just giving you, here's what chicken turtle genomic diversity looks like, well, I'll give you a couple of other species too. And I want to start with pupfish because we just finished a big study on these. And so the white sands pupfish, genus Cyprinodon, is endemic to the Tularosa Basin in New Mexico. And the international organization IUCN, which publishes the infamous Red List, finds that they are or categorizes them as endangered. They're found in only 2 springs and 2 small streams in this desert environment, and those populations constitute 2 different ESUs, or these, again, evolutionarily significant units that were defined not by geneticists, but by real fisheries biologists, right, who were looking at the differences in salinity in these water bodies, the parasite communities, the morphologies of the fishes, and said, look, we think we've got 2 different evolutionary significant units here. And they're subject to, to some threats to their survival, their thriving, their long-term prognosis, right? They've got some threats there that have to be addressed. And while that's being done, then we were funded to look at some of the genomics. And so to put this in context, of course, all of you in Texas have some idea where New Mexico is, but in Europe, they did not. So I use this slide there, but here's the Tularosa Basin, right? Just north of White Sands Missile Range is where our study sites are. And you can see in blue here some of the different populations. And I'm going to use about 2 or 3 slides to talk about a couple of years worth of work. So forgive me for jumping right into some of this. But the first measure I want to talk about is individual heterozygosity. That is, for an individual fish, what was the heterozygosity across its genome? And what you can see is that there are some differences. Here I've got ESU1 in blue or dark blue or light blue, and then these other colors represent different populations at different time points in the other ESU. And you can see that ESU1 has got quite a bit more genomic diversity than ESU2, right? It's probably 2 to 3 times as much diversity on average. So the point is that the genomics can differentiate between these ESUs just on the basis of the diversity. But when we use a statistical technique called a principal component analysis of these whole genome sequence data to try and plot the similarity of these fishes, where each individual dot here, each different point represents a single individual fish, a couple of things emerge. One is that the two ESUs, which are different colors, separate from one another very well. The x-axis shows that about 45% of the variation um, in this data set can be attributed to this principal component. Another thing that comes out of this is that one of these populations, the black circles, is actually a, a population that was established by humans artificially in a different water body from the source population, which is the X's here. And what we see is that genomically they're super similar, as they should be, because one population was established from the other. And both of these are much more similar to each other than either is to the blue population. And so I think this slide really nicely illustrates the power of these whole genome sequences to delineate the two different ESUs. Okay. We can also use these data not just in simple similarity, but looking at the phylogenetic relationships of all the individuals. And the take-home points here from these two phylogenetic trees are, these are, if you want to think about it this way, these are to some degrees the extended pedigrees of all the individuals in the study. And what we see is that the blue ESU1, which is both the light blue and the dark blue, they're just different time points. Those all have a single common ancestor, right? And likewise, all of the other individual sample from all of the other populations have a single common ancestor. And we see the same pattern at the mitochondrial genome as well. Even though there's just too much data to show here, we have gone through this very carefully, and that is what's happening here. So the phylogenetic trees also delineate these ESUs quite dramatically. And so in terms of the pupfish, what we found with the population genomics is that they very strongly support the delineation of the two ESUs based on salinity, parasite communities, fish morphology, and so forth. What I didn't show you is all these formal analyses of species delimitation that we did that consistently identified two different species that correspond to the two different ESUs, and that we think formal taxonomic descriptions are warranted. So instead of the white sands pupfish, we think there should be another species named that corresponds to the two different ESUs. But this is, of course, not always the case. And so I'll talk about a very different biological system, the genomics of prairie chicken here. And one of the things that I'm going to go back for a second, because I forgot to mention this, to look at this geographic scale. So here, the difference between the Malpais Spring ESU and the others is we're just talking about, 5, 10, 15 miles. It's a very small geographic range, but a very large genomic divergence between these pupfish ESUs. Well, with the prairie chickens, we're talking about two species, the greater prairie chicken, GRPC, which will consistently be, I hope consistently, in gold throughout the next few slides, and the lesser prairie chicken, which will hopefully be the LEPC in red. And it's red because this species is endangered. And the greater prairie chicken is not. It's hunted recreationally in parts of its range. But of course, they both face habitat loss and fragmentation of our short and long grass prairies. But this occurs on a much larger geographic range. So historically, you can see that greater prairie chickens were found from the Gulf of Mexico all the way up into interior Canada. Today, it's a much more limited range, but throughout the upper Great Plains. The lesser prairie chicken has an even smaller range. Um, but some of this is also in the Great Plains and the Panhandle of Texas. Texas does not have graders, but it does have lessers. And as I showed here, keep in mind that these have already been designated two different species by ornithologists. So, because of a lot of different reasons, um, associated with the U.S. Fish and Wildlife Service listing of the lesser prairie chicken as deserving of protection under the Endangered Species Act a couple of years ago, we were involved in this study of um, hundreds of prairie chickens, sequencing their genomes, focusing primarily on the lesser prairie chicken, but we also had a couple of dozen greater prairie chicken samples. In here, this figure, I think, nicely illustrates 2 points. One are the physical differences and similarities between greater and lesser prairie chickens, but also the fact that they both occur on the same breeding grounds at the same time of year, and that ornithologists through binoculars see these interspecific matings. They see hybridization. What was less clear before our study is whether genes actually made it from species, from the greater into the lesser or the lesser into the greater, or whether they ended up in sterile offspring, right, that maybe were infertile, in which case it would sort of be a genetic dead end. And so our study addressed this. And when we look at delineation in the context of prairie chicken phylogenetic trees, both the nuclear tree and the mitochondrial tree, what we see is that there is no consistent delineation like we saw of, the whole blue branch on the tree in the pupfish. There's nothing like that here. And both species are scattered all the way around, even though it's hard to see because there's so many data points on here. Again, we've zoomed in, gone through this in detail. And the point is, there's really no groupings of distinct species or ESUs, or to use Fish and Wildlife Service language, the distinct population segments are not nearly as clear when we're talking about both these species at once. And so here's our map again. If we zoom in, you can see here is the gold. And again, the scale, here we're talking hundreds of miles across the scale. The gold represents the current range of the greater prairie chicken. The red is the range of the lesser prairie chicken today. And Fish and Wildlife Service has designated this line that separates the northern um, threatened population of lesser prairie chickens from the southern endangered population of prairie chickens. So when we sequence their genomes, these are how the data fell out. And when we look down here first at this principal component analysis, we can see that it more or less it did group them into two discrete groupings, but it's not necessarily due to the two different species like we expected, where the graders would be in one group and the lessers in another group. But instead, it's more based on this geographical boundaries, and shows that the southern population of lesser prairie chickens is quite distinct from the northern population of lesser prairie chickens. And when we compare these to greater prairie chickens, we see that there's a lot of ambiguity. And that's shown both here in the PCA, where we've got gold individuals in here with red, and vice versa. But we've also got all these intermediate individuals, and these are zooming in on the genomes of these individuals, and we can see that some of them are about 50-50, whereas some are 75% one species, 25% the other, or vice versa. And so another way to think about delineation is about these admixture. And this top figure here shows admixture proportions in individuals, where each column is an individual bird. So this is for whatever, a dozen individuals. Here we have the same thing, but shown for hundreds of individuals, where each of these little thin columns represents an individual bird. And so this figure shows the individual's affinity to a particular gene pool. All right, and we start with the possibility of K equals 2, where K is just the number of populations, or the number of delineation units. Uh, and here we would say this group of birds is mostly the blue genome. This group is mostly the tan genome, etc. And here's where we would have three different ones, four different groupings, five different groupings with the same number of birds. And this approach is totally ignoring the geographic location information associated with each individual. So it only has the genomic information to try and put everything together. And I'm not going to go into the details of how it works, but right, everybody's familiar with the mathematical model of E equals mc squared, where energy is equal to the mass of a particular um, of a particulate, a matter, times the square velocity of light. Um, we're using a similar mathematical model. It's a very simple model based on Harding-Weinberg equilibrium, and we're telling a computer algorithm to take all these genome sequences and put them in these different categories of 2 populations, 3 populations, 4 populations, 5 populations, and tell us what the best fit is using a mathematical model. And when we do that, it turns out that with these prairie chicken data, the best fit model by far is that K equals 2, right? Which would be this top scenario. And you might think, well, that makes sense, right? There were 2 species, it should put them in those 2 bins, but that's not the case. This white line actually differentiates greater prairie chickens to the left versus lesser prairie chickens to the right. And what we see is that many of the lesser prairie chickens have a more similar genome to the greater prairie chickens than they do to the southern um, population of lesser prairie chickens. You can see the geography, which was put on this figure after the algorithm did its work. And so the northern and southern populations are quite distinct from one another. But the northern population of lessers is very similar to the greater prairie chickens. And if we tell it to make K equals 3, you can see in the northern lesser prairie chicken population all this introgression of greater prairie chicken genomes into them. So here's an individual that's about 50% one species, 50% the other species, and then we see others that maybe 25% one species, 75% the others. And when we look at the greater prairie chickens, we see the same thing. There's some of this leakage of the lesser prairie chicken genome into the greater prairie chicken genome. Okay, so um, to put this in context, prairie chickens have pretty normal levels of genetic diversity, genomic diversity. It doesn't seem to be changing over the last 10 years or so. We see no evidence of inbreeding. The population subdivision in these lesser prairie chickens is quite modest, and it's mostly between the northern and southern population segments. Um, and there's more differentiation between them than there are between the 2 different species. And the genomic data showed clear evidence that the hybridization that's observed in the field actually leads to this process of introgression, the leakage of genes from one species into another, and it works both ways, lesser genes going into graders and greater genes going into lessers. And this is a complicated situation with the fact that one of these is, well, up until very recently, a protected species under the Endangered Species Act, whereas the other could be a hunted species in portions of its range, and there's hybrids. And I'm not a lawyer, but I think there's probably a lot of legal ramifications to that. And so in the end, this study showed that the binomial taxonomy, trying to put these birds into 2 simple buckets, black and white, doesn't really accurately reflect the biological reality. And I think it's interesting that before we got to 2 different species, the ornithologists used to call these super species when you consider the other prairie grouse as well. So that's a little bit of background. I'm sure some of you are thinking, what the heck? I thought this was going to be a turtle talk. And it is, I promise, only to talk about turtles the rest of the time. But I really did want to give some context and some examples for some of the approaches like the phylogenetic trees or the principal components analysis, etc. So I am going to talk about turtles now. This gets back to Natalie's chapter on whole genome sequencing, trying to provide insight into population structure and life history for conservation and management of these really fascinating organisms. And there's a big team of people that you can see that are working on these. So, chicken turtles are in the family Amididae, which is somewhere in the neighborhood of 50 million years old. So this lineage has been around for a while, and this particular genus, Dirocheles, has been around somewhere around 25 million years ago, emerged in the Cenozoic. Chicken turtles are native to the southeastern United States. They inhabit shallow or slow-moving bodies of water that have a lot of emergent vegetation, muddy substrates. They're carnivorous, semi-terrestrial, and maybe endangered, at least the western subspecies, and I'll come back to that in a bit. They can be pretty long-lived, up to 20 years. And I've been told that they got their name because they taste like chicken, and I've got no evidence for that. The three different subspecies are shown here, and I'm going to talk almost exclusively about the western subspecies today. But I'll show you the taxonomy here of the other two subspecies, the Florida and the Easterns, because the next slide I think I'll show you a map. It shows here's the range of chicken turtles, right? And so the Florida subspecies is on the Florida Peninsula. Um, the eastern chicken turtle is in the other southeastern part of the United States. The western chicken turtle occurs west of the Mississippi River, as you can see here. So here's a map that shows some of Wade and colleagues have done some work on species distribution modeling and trying to look at the probability of occurrence based on habitat for chicken turtles. And you can see the maps here of the western chicken turtle. All of our samples for this study come from Texas. We don't have any samples from Oklahoma, Arkansas, Louisiana, or down here in southern Missouri. And part of the reason we're interested in this is in 2011, the U.S. Fish and Wildlife Service published this partial 90-day finding on a petition to list a bunch of species in the southeastern U.S. as endangered or threatened. And so they said at this time they were initiating a status review of many different species, including the western chicken turtle. And as of last week, this is still under review, according to Fish and Wildlife Service. And so we've been working with the state of Texas to provide them with some of the, what I hope are insights that have emerged from the genomics work to help them as they move forward with the management and conservation of this species in light of this ongoing status review. So the conservation concerns around western chicken turtles are largely due to the fact that their numbers are thought to be in decline. And a lot of that, I'm sure, is habitat related, right? The Houston area has the most high quality habitat in some regions, but urbanization around Houston, right? Most everybody on this webinar is going to appreciate that urbanization's a big problem. And the wetland loss diminishes habitat availability, which of course probably limits western chicken turtle numbers. But from a genomic perspective, it's also important to note that this urbanization and wetland loss leads to a patchwork habitat that can also reduce connectedness of the habitat, which influences genomic diversity indirectly. And so I've used this term a few times, genetic or genomic diversity. I kind of use those interchangeably, but it's the idea that a diverse gene pool, like shown on the left here, is what we see in large populations compared to isolated smaller gene pools shown on the right, where we have less genetic diversity overall, but particularly within populations. The situation on the left with a large gene pool leads to more adaptive capacity and a potential for long-term survival that's significantly higher than we see over here on the right, where we've got lower adaptive capacity. And the diverse gene pool has more ecological resilience, the ability to withstand environmental change than any of these populations or subpopulations on the right that have less resilience. So this is why we're interested in genetic diversity. And I think one of the things I always like to point out about genomic data is that it really provides powerful tools to study species that can be hard to follow around and track. Things like western chicken turtles, which are tough, right? Due in part to these long estivations where they burrow underground, as you can see in the bottom photo here. This leads to low detection rates. So if you have chicken turtles, it can be hard to detect them, and low capture rates. You can go out and put out a lot of traps, spend a lot of person hours in the field, and get little return on that investment. And I think that Natalie's study has showed that population genomics can help a lot in this regard. Um, and Natalie did get to spend some time out in the field and catch some turtles with Toby and Wade and collaborators, and stomp around Southeast Texas in the heat of the summer. And she caught lots of species, lots of herps, but she caught no chicken turtles. And that gets to the point that I think she was in the field a week or so with these guys. And that's a lot of person hours when you multiply all that together. So we did not collect these samples. They were collected by teams of, expert herpetologists who were actually out trying to collect Western chicken turtles, or in some cases, opportunistic roadkill. Find one on the side of the road, and that dead animal still has DNA that can be retrieved and sequenced. And so Natalie did some of that as well. We had 8 different sites here across southeastern Texas, across a few hundred kilometers. As you can see, 150, 200 miles at most, about 10 or 11 samples per site on average. And then Natalie started by sequencing the genome, and then followed that up with some individual, what we call resequencing across the rest of the 88 individuals. And then did a number of comparative analyses to try and sort that all out. And so it turns out the genome is about 2.2 billion nucleotides in length. So that's all those GAs, Ts, and Cs strung together. It's a long string. And if I did the math right, the resequencing data set is about 400 times that. So it's an even much bigger number. So she has wrangled lots of GAs, Ts, and Cs so far throughout her dissertation. And so there's 3 different things that we've really looked at here, and they include population structure, um, and then migration surfaces to try and identify landscape barriers or filters to gene flow, and then overall genomic diversity, which, as I mentioned earlier, is some indication of population health and adaptive capacity. And so the population structure looks like this. So we got our sampling sites over here on the left with the sampling sizes above those. And then the principal component analysis, you can see here, just like with the pupfish, it does a pretty remarkable job of being able to take an individual turtle's genome and compare it to everybody else, and then assign it to the same geographic sampling site where it was collected. So this approach, just to reemphasize, does not use any geographic information, right? This is based solely on the DNA sequences. So all these purple individuals that were sampled at Gordy are much more similar from a DNA sequence perspective to one another than any of them are to these individuals from, say, Wharton over here, or from Allison Bayou down here. And similarly, all these from Allison Bayou are much more similar to each other than any of them are to those from these other sites. And of course, a couple of these sites, we only had 1 or 2 individuals from. So this is a limitation of working with species that are really hard to detect and sample in the field. So this really pronounced population structure means things like, if we have a roadkill sample, it's if Paul Crump brought us another sample tomorrow, we could sequence the genome, look at this, and with a very high degree of um, certainty, assign it back to a source population. And that's what this figure shows, that the individuals that were sampled from Alazan Bayou were assigned back using this algorithm to Alazan Bayou with 100% certainty, or 100% of the time. And similarly with Wharton and Warren, it's a little bit less in a couple of the other populations, but we also had some of these populations are really close to one another. So the Buller and Warren populations are only like 5-6 kilometers apart. I'm sorry, 5 or 6 miles apart. So it's a really small geographic scale, but there's a tremendous amount of resolution in the genomic data. So that was all based on the nuclear genome. This is the mitochondrial genome showed over here, where the different colors in the pie represent different mitochondrial haplotypes or different mitochondrial alleles. And we see that even in populations that are very near one another, we see different haplotypes present at different frequencies. Um, and so we see a lot of diversity within populations, and there's a lot of variation among the populations in the mitochondrial genomes. And when we look at the nuclear trees, we see what I think is a remarkable amount of structure in the nuclear tree. So this is much more reminiscent of the pupfish than of the prairie chicken, where all these individuals up here at the top share a common ancestor of this red clade. We see the same thing with a blue clade down here, a purple clade. And it's not perfect. We see, an occasional what we might call a misassignment here, and that could be due to some lack of resolution in the genomic data, but it could also be the fact that maybe this individual was sampled in one population, but it was dispersing from another natal population where it was actually born. We don't know the answer to that, but overall, there's a remarkable amount of structure here. These colors are not all mixed up. A similar situation occurs with the mitochondrial tree as well. It's not quite as pronounced, but almost. So a lot of structure there. When Natalie and another of my students, Zhang Yun, did the themes analysis to try and get at this migration surface, where they used the genotype data and the spatial coordinates of where the samples were collected to try and identify any barriers to gene flow or filters to gene flow. This is what they got. And if you're not sure what you're looking at, this is a map of Texas. I think this is Galveston Bay, our sampling sites. And if it all looks white, that's because there's no apparent barriers to gene flow. If there were, you would see the colored um, schemes here, and they really don't seem to exist. So when you put together some of this genetic genomic structure analysis, our principal component analysis, which clusters samples according to similarity, shows very strong population structure. The mitochondrial analysis shows strong structure. The phylogenomic trees, both nuclear and mitochondrial, show really strong population structure, but the phemes analysis shows no barriers to gene flow. And so that's made us pause and think about this, because population structure is often driven by barriers to gene flow. And there's a million examples I talk about in my conservation genetics class. One, in Arizona, mule deer populations on the north and south rims of the Grand Canyon are differentiated in part because it's a barrier to gene flow. They don't go from the north to the south side of the Grand Canyon. Timber rattlesnakes in New York are differentiated on either side of a major highway because it's a barrier to gene flow. And even bird populations in the Amazon in South America, populations are differentiated across either side. So population structure is often driven by barriers to gene flow. We don't see any here with the chicken turtle, which begs the question, why so much population structure? And I'm going to leave you hanging there for just a couple of minutes. And I'm going to shift gears and talk about genomic diversity, and then I'm going to come back to that question. But the genomic diversity that we see in the western chicken turtle is, it's similar to what we see in the pupfish and sorry, similar to what we see in the lesser prairie chicken. Um, and to put it in context, it doesn't seem to be too remarkable in terms of all these other um, genomic diversity estimates that are available from other turtle species. But when you look closely at how these species are categorized by IUCN on their red list, the species that are in red or orange is here are those that are most dramatically at risk. And the chicken turtle seems to have genetic diversity levels that are more similar to them than some of the more common species like eastern musk turtle or ornate box turtle. When we look at genomic diversity in space, what this figure shows is that the darker greens are more um, genomic diversity, and that tends to be more in the eastern parts of the range and less in the western parts of the range. And I think this makes good sense because if you think about sort of the core of the species, not just the western chicken turtle, but the whole species, or even if you're thinking about just the western chicken turtle, if you looked at sort of the centroid of the distribution here, what you see is that it sort of makes sense to me that there's more genomic diversity in central East Texas than in on the periphery of the range. This is more in the core of the range of the species. So I think that makes sense. And one of the most interesting things I thought about this study is that Natalie found that, some of these sampling sites have similar levels of genomic diversity, maybe some subtle differences, for example, between Gordon and Warren, but others, this Wharton site has significantly different lower levels of genomic diversity than do many of the other sites. And this is consistent with what we saw all the structure in the phylogenetic tree and in the PCA, because if there was a lot of gene flow between these sites, we would see much more similar levels of genomic diversity among all the sites. Um, yeah, in this heterozygosity is about 3 times that of prairie grouse and about the same as the pupfish. So I'm going to wrap things up over the next couple of slides and try and drive home the major points as I see them. And so the assembled genome of the western chicken turtle is about 2.2 billion base pairs. Our survey of population genomic diversity revealed clear structure and high confidence in population assignment using a variety of different approaches, but we detected no extrinsic barriers to gene flow. The genomic diversity values varied among sampling sites, and that's just what the prior slide was. And there's low genomic diversity compared to non-threatened turtle species. In other words, it's similar genomic diversity to threatened turtle species. And all this population structure seen in the PCA, the phylogenomic trees, the mitochondrial frequencies, the assignment tests, this seems to be due to intrinsic factors. And what do we mean by that? And I think this is the idea of natal philopatry, returning to or remaining in one's birthplace to reproduce. This seems to be the biological phenomenon that's most consistent with the genomic data that we have. Um, and that's consistent but imperfect fidelity to natal wetlands. And so, I mentioned earlier that there may be um, some dispersers or some straying individuals from one population to another. And I think our data are very consistent with that. So if Western chicken turtles reproduced in their natal wetlands, the gene pools are differentiated not because of extrinsic barriers to movement, but due to intrinsic drivers for the turtles themselves. And I think this is a bit analogous to the situation almost everybody's heard of natal homing that occurs in salmonid fishes in the Pacific. They forage in the ocean and then navigate back to their natal streams for reproduction. And I can imagine something much like that happening with the western chicken turtle. They may move across the landscape in telemetry studies, but the genetic data suggests they come back to the same wetland where they were hatched to reproduce. And this is not an idea that is unique to us. This is seen in other turtle families. So Natalie did this work across different turtle families, and you can see the closed circles here mean that's a family where natal phylopatry has been documented. Open circles means no evidence of natal phylopatry. So it happens in some families, not in all. And she's here categorized the chicken turtle family as having evidence of natal phylopatry in at least one species, being the western chicken turtle. Okay. So, so what? Why does this matter? Um, well, one question is, why do western chicken turtles exhibit phylopatry? And we don't know for certain the answer to that. The previous slide doesn't suggest that it's not phylogenetically conserved, right? If everybody on this branch was um, phylopatric, and all of these would be filled, but that doesn't seem to be the case. So it doesn't seem to be, um, you know, uh, phylogenetic. So Natalie's thinking is that maybe it's variation in temperature and precipitation, make suitable habitat unpredictable, and coupling that with the estivation strategy may disincentivize movement of turtles to, explore alternative breeding habitat. And why this might matter includes the fact that new breeding habitat might be colonized very slowly when western chicken turtles are faced with environmental disturbance. So if they're breeding in one wetland and a developer paves over it for a parking lot, then obviously they got to find a new wetland, and that may not go very well for species that have natal phylopatry. But furthermore, chicken turtles likely have temperature-dependent sex determination, which I didn't go into today. I'm not going to because of the time. Uh, but many turtles have temperature-dependent sex determination, so that the sex ratio in the offspring in a clutch of eggs is determined in large part by the incubation temperature. And so strong natal fidelity could hamper adaptive response to climate change by skewing sex ratios. So some of the insights that I think have emerged are lowish genomic diversity in western chicken turtles, clear population structure, and high confidence in population assignments, which, as I mentioned, could be useful for identifying migrants or dispersers. Um, Very fine scale differentiation is apparent, even at very nearby um, geographic sampling sites. And I think the most reasonable biological explanation is natal phylopatry, that adults return to or remain in their birthplace to breed. And I think hopefully all this should be considered as part of conservation and management moving forward. So, overall, I think population genomics can be very effective at helping to delineate conservation units, including species, populations, individuals, ESUs, DPSs, and so forth. And it can do this while delineating, can also reveal key biological insights. And I haven't talked about a few of these. Talked a little bit about bidirectional integration, but especially natal phylopatry in the case of the turtles. And so I will stop there, and hopefully we've got a little bit of time for questions, but I'll defer to Paul's judgment for that. And so I thank everybody very much for your attention.
Yes, sir. Thank you, Andrew. We definitely have plenty of time for questions. We've got about, 8 or 10 minutes for the end of the session, so please post any you may have. There's none in there currently, but I have a couple of questions. I think about it, Andrew, our job is kind of, the management of these populations in the wild. And so thinking about how you would design a conservation strategy for a species that displays a phylopatric pattern of kind of reproduction versus a more, isolation by distance type pattern. And my immediate reaction is that we should be placing sort of conservation emphasis on areas immediately surrounding wetlands then, right? Protecting upland habitat, thinking about management actions in those strategies versus, thinking too much about connectivity between populations.
Pretty easy. I've told this to, all my students. Natalie and Jong Yoon have probably heard me say this, right? The hard part is trying to figure out how to put this in a context when you've got, like TPWD is charged with managing all the wildlife in the state, and the species differ so dramatically. Everything from isolation by distance to strong natal phylopatry to just sort of this random dispersal that some species do. And I think it's a huge challenge to think about how to our job, I think, is to work with managers like yourself, conservationists, to try and put the genomic data into context, which is why I talked about the pupfish and the prairie chicken as well, and not look at it in a vacuum. Um, because I think that would be a very bad idea with these chicken turtles, because I think one could make the argument, I would not make it, but I think you could make the argument that, well, every one of these wetlands that you sampled is going to be different. So they should all be managed separately. And I think that would be an administrative nightmare. Um, so I don't suppose that really answers your question, Paul, but um, maybe if you can rephrase it, I could take another stab at it.
Yeah, I'm just thinking about how sort of the application of limited conservation resources to
Yeah.
focus on. Like I think of some of the species, we worry a lot about dispersal, right? Population persistence at a site requires the free exchange of individuals across the landscape. So we think about things like wildlife crossings and
Hmm.
the quality of the habitat matrix. For chicken turtles, maybe then the emphasis should be put on um, you know the upland habitat immediately surrounding either wetlands or wetland complexes as a priority versus worrying too much about movement across the landscape at larger spatial scales.
Yeah, I couldn't argue with that.
Yeah.
I think
Okay.
that's very reasonable.
Okay. Some of the questions are coming in. I think I maybe had the saying set up wrong, so I corrected that. They're flying in now. Okay. It's a question here. Does the fact that there are no barriers to genetic flow make the species more or less hardy in terms of survival?
Oh boy, that's a tough question. That's a good question. More or less hardy in terms of survival. Um, Gosh, that's a tough one. I wish Natalie was, I wish I could just give this one to her. This is like a PhD defense question. Um, I don't know. I suppose if you view it from, eh, I would view this from a long time scale, right? And the chicken turtles occur across the southeastern U.S. Um, and if this same pattern exists in the Florida subspecies and the eastern subspecies, and I bet it does, and if you saw this all across, it would argue to me that maybe this is a good strategy, right? Long term, they've been around for many millennia. And so I think it served them pretty well so far. And I guess the idea would be that each site is a little bit insulated. So if one blinks out, well, we've got all these other ones that sort of serve as backups. And I don't know if that's the right answer, but that's uh, that's my response.
No, understood. Okay, so somebody here was wondering if you have data to compare the level of structure of phylopatry with that of the eastern chicken turtles. Any comparative stuff?
Yeah, I'd love to have that, but the answer is no. We don't have anything from the eastern chicken turtle or the Florida chicken turtle, but I'd very much like to, because yeah, I like the Mississippi River. Is it a barrier or a filter? I don't know, but it would be interesting.
So, how can illegal collection or reintroduction from the pet trade affect the genetics of populations like this? Seems like people's obsession with captivating wild turtles could really add some outliers to the data set. Oh, I see what the question's about, like humans moving animals around in the
Yeah.
wild.
Yes, absolutely right. So, there was a little bit of there was a little bit of uncertainty in the data sets and the population assignments and all, and a couple of individuals in the phylogenomic trees where I said, well, maybe they're dispersers, maybe that's natural, right? That a few individuals don't come back home to breed, but maybe it could be a signature of humans having moved individuals around. I don't have any idea.
Yeah. So somebody here comments, I've been working with Western chicken turtles in Arkansas for the last couple of years. Would you have any interest in including the population I've identified in any genomic studies?
Uh, I'm not quite sure I got all that, Paul. Could you repeat it?
Oh, yes, sir. Somebody commented that they've been working with Western chicken turtles in Arkansas for the last couple of years and asked if you'd have any interest in including, I guess, samples from the populations that they've been working with in this kind of work.
Yeah, potentially. I guess I would say, my email's dwoody@purdue.edu. So, if you send me an email, we could follow up on that. Some of it'll come down to our funding situation, but maybe, 100 individuals that no, we can't afford that. But if it's a few, maybe.
Okay, that's good. There's some sort of ecological questions here about can we talk a little bit about the chicken turtle's purpose and method of burying itself for long periods? Um, this is, my understanding of this is that it's an adaptation to the sort of ephemeral nature of the wetland habitats that they use. And it's a common thing across all of the subspecies across the southeast, from what I understand. So it's probably an adaptation to drought and
Hmm.
and other inhospitable conditions.
Yeah, I think it's the same with the pupfish in New Mexico, by the way. They're thought to be able to estivate to some extent.
Really, wow.
Yeah, for similar reasons.
This the last question here kind of hints at another question I had too. Do males and females return to their birth wetlands to breed and lay eggs, or just females return to lay eggs after breeding elsewhere?
Yeah, I think that's a great question. And I think that, our data suggests that it's both, right? It's both males and females because it would lead to very different structure. We would see differences between the mitochondrial trees, which are just based on the maternal lineage, versus the nuclear trees, which is maternal plus paternal. And because they're very similar, we think both sexes are um, exhibiting natal phylopatry.
That's great. Okay, now one last question has popped up here before we sign off here. To what extent can genetic divergence in Western chicken turtle populations seen today be explained by the relative recent changes to habitat and contemporary barriers to movement, such as roads, development, loss of wetlands? In other words, would you still see distinct genetic islands we see today if none of the changes to the landscape had happened over the last 100 years or so? It's a long question.
The things we wondered, that's one of the reasons why we came up with the migration surface, right? Is to try and get at this idea of, our roads, major barriers, or, these river. I mean, when Natalie first started on this, we, tried to partition all our populations into river drainages and try and figure out some other these extrinsic factors that could play a role here, and none of those emerged as a smoking gun. The only thing that I think really explains the data is the intrinsic, the turtle specific natal phylopatry that they come back. It does not seem to be anything either historic like river drainages or contemporary like highways.
That's great. Well, that's the last of the questions. Andrew, thank you so much again for joining us today. This is a great